We are working from the February-March 2018 paper. It's paper 22, so it is the extended multiple choice questions. And we are going to hit whatever topic we get. So question 1, way back at the beginning of the course, biology is the study of living things. Which characteristic applies to all forms of life? Okay, so characteristic A, able to move from place to place. Well, it's a characteristic of life, but not all forms of life. Able to reproduce. Well, that sounds suspiciously like it is everybody, but let's just double check the other answers as well. Carry out photosynthesis, not all of them. And possess a nervous system, again, not all of them. So we have confirmed, we're completely happy with answer B. And the thing is, as you work through your final exam, you can work through this like this as well. You can scribble all over your question paper. Just make sure that your answer sheet for the multiple choice is lovely and neat and tidy and just has colored into your final answer. Okay, question 2. 2 animals have an identical sequence of amino acids, in one of the proteins found in their cells. What does this indicate about these animals? Now remember that the sequence of amino acids depends on DNA. So if they have an identical sequence, then they have more similar, well, they have identical, not even just more similar, they have identical DNA for this protein. So when we think about DNA and we think about DNA being identical, we're thinking about the fact that these organisms are very closely related. So we can work through now their answers. They've been eating the same types of food? No, I don't think so. They have not been exposed to substances that cause mutation. Well, mutation changes the DNA, so if they haven't had mutations, then they would have the same DNA. So let's leave that one for now. They must be members of the same genus. Now, we're saying they've got identical DNA, so we're saying they're actually probably more closely related than a genus. They share a recent ancestor. Definite. Yes, that one we like. We can confirm that B is not what we're looking for, and D is most definitely the idea. Because if they're identical if their DNA is identical, then they've got to be very recently linked in order to have that same amino acids coming from that same DNA. Question 3. Sticking with classification, when we've got a question like this, we know straight away we are looking at a dichotomous key. So, the diagram shows part of a flowering plant. Using the key, identify this plant. So the first question says, do we have 3 petals or more than 3 petals? So if we look at a flower there, we've got 1, 2, 3, 4 petals. So we go to 3. Then we look at 3 and we say, are the leaves parallel veined or not parallel veined? So we look at a leaf. And we can see that the veins are branching. Okay? If it was parallel, they would be parallel, like parallel lines. So the leaves are not parallel veined, so the answer is D. So you're systematically working through the information that's given in the question so that you can get to the final answer. Everybody happy? So, something different. The diagram shows a single cell from an organism called Spirogyra. Now, you haven't studied that, but it doesn't matter, okay? This is what we call an applied question. So it's going to make you use the information that you've got and that you've learned in order to answer this question. So we see a vacuole, cytoplasm, chloroplast, cell wall, cytoplasmic strand, and nucleus. Okay, which features does Spirogyra share with plant cells? So of these features here, what do plant cells also have? Do plant cells have a cell wall? Absolutely. Chloroplasts? Yes. Cytoplasm? Most definitely. Nucleus? Absolutely. and a vacuole, definitely permanent large vacuole when it comes to plants. So we've answered independently. Now we go and we see which row in the table matches all our ticks, and there it is. A is the answer that matches ours. So wherever possible, we want to be answering these questions, then looking for the one that matches, rather than getting led astray and distracted by dodgy, weird answers that they might put. Okay, so the next one. A student was told that a drawing of a bacterial cell had been magnified 30,000 times. The length of the drawing was 45 millimeters. What was the actual length of the bacterium? So magnification can come up in our, in our theory papers as well as our alternative to fact papers. And the thing that we've got here is we've got a 45 millimeters. Now, when it comes to bacteriums, what do we actually use to measure them? We don't use millimeters. We use micrometers. So we need to get out of that 45 millimeters into micrometers before we can then calculate the actual length. So that's why we measure in millimeters always, because we can say 45 times 1,000 equals 45,000 micrometers. Then we need to think about magnification is image over actual. So then if we want to get actual, we would say, well then, let's do our triangle as well, seeing how different people like different things. So from the triangle or from rearrangements, we know that actual equals image over magnification. So in this case, it would be 45,000 over 30,000. Well, all of those zeros are going to cancel. 45 divided by 3. Where I've missed something, I've missed a 0. That's 30,000. Yes, 45 divided by 30, that sounds a whole lot better. So our answer then is the 1.5 micrometers. So always remember, when you're working with magnification, you must have things in the same units. Any measurements must be in the same units. And when you're working with very small measurements of cells or smaller than cells, you always want to be working in micrometers. Millimeters is what we use when we're measuring our images or the diagrams that we've drawn. Question 6. Which graph represents the effect of increasing temperature on the rate of diffusion? And we don't even look at the graphs. First of all, we think what happens when we increase temperature? We're going to increase diffusion because we're increasing that kinetic energy. So if we're increasing both, our graph should look like this. And then we go and we look at the graphs we're given, rate of diffusion against temperature, going up to the right, and the answer is D. So again, we're using our knowledge to answer the question and then finding the matching correct answer. Question 7. Protoplasts are plant cells that have had their cell walls removed. What happens if plant protoplasts are placed in distilled water? They get larger or they get smaller? Now we need to think again, because here we are talking about osmosis, right? Distilled water is pure water. So this is very high water potential. When you have high water potential outside, what happens? There's your cell. The water is going to move in. So our cell is definitely going to get bigger. Now, normally when we talk about plant cells, when the water goes in and osmosis happens. Now the contents get bigger, and then we get turgid and we get pressure and all of those wonderful things. But that is all dependent on that cell wall. So if there's no cell wall, then our plant cell is going to act just like an animal cell. It's going to get larger and larger and larger to a point where you can't hold that pressure. And then it's going to pop. So our cell, our plant protoplast, will get larger, but then it will burst because it has no cell wall to hold it safe with that pressure. Question 8. When a food substance is tested with iodine solution, which color shows the presence of starch? So remember, iodine solution is a yellow-brown color to start with. Sort of orangey color, okay? Positive is blue-black. So the answer is A. You must be familiar with the tests for all of the different biological molecules because, again, they can come up in the theory as well as in the alternative to practical. They're not restricted to that paper 6 only. Question 9. The graph shows the activity of 3 digestive enzymes at differing pH levels. Okay, so we've got enzyme X, enzyme Y, enzyme Z. pH is from 1 to 12, and we're measuring enzyme activity. So you can see for each enzyme, activity goes up and then it goes down. So it's showing the optimum pH is different for each of these enzymes. Which statement is correct? But we're going to have to work through each of these and decide yes or no. Enzymes X and Y are both active at pH 7. So pH 7, that cuts through Y and Z, not X. So that's wrong. Enzymes X and Z are both active at pH 4. pH 4 cuts through Y and X, not Z. Enzymes Y and Z are both active at pH 4. Again, Z is not active at 4. Z only starts at 4.5. So let's see if they get it right the last time. Enzymes Y and Z are both active at pH 8. So coming up from pH 8, and there we hit Z and we hit Y. So finally they got a correct statement. Both of those enzymes are active at pH 8. From enzymes to plant cells. Okay, so the diagram shows a cross-section of part of a leaf. We can see we've got the midrib, the lower epidermis, the upper epidermis, and then a layer X. Layer X, right underneath the upper epidermis. This is our palisade mesophile layer. Which type of cell is found in layer X? Well, palisade cells. And the really special thing about those palisade cells is that long, skinny shape with lots and lots of chloroplasts. So if we look at all of these cells, the best option for that would be option A. Question 11. Which molecule contains magnesium? Chlorophyll, fat, glucose, or starch? Well, if we think about plant nutrition, we get a little bit of information about magnesium. If a plant is deficient in magnesium, then it is not a proper green color, is it? Because it can't make chlorophyll. So the molecule that contains magnesium is chlorophyll. Question 12. In which part of the body of a mammal does mechanical digestion occur? Mechanical digestion, all about the breaking down of pieces of food into smaller pieces of food no chemical change. The biggest culprit for this is your teeth, of course, and the teeth are found in the mouth. So mechanical digestion is not occurring in the gallbladder or the liver or the pancreas. It is occurring in the mouth. Question 13. During growth, potato plants produce flowers and underground storage organs called tubers. During this time, which parts of the plant act as sources and sinks for translocation? Okay, so a source is where photosynthesis is occurring. A sink is a storage or use site. Okay, somewhere where those products of photosynthesis are either being stored or used. So if we think about where does photosynthesis occur? Photosynthesis is occurring in the leaves. So these guys are our source. Potato tubers are very much a storage organ, very good for us. Okay, so those guys are a sink. And flowers, also, they're not photosynthesizing. Okay, they're being using these products of photosynthesis to grow the flower. Okay, so they also act as a sink. So we have sink, source, sink, and the answer that matches that is B. Flowers are a sink, leaves are a source, potato tubers are a sink, while the plant is actively growing. Question 14. What is an advantage of a double circulatory system in mammals? Blood can flow down the body on the left and up the body on the right. I don't think so, because if that were true, then our right foot would never get any blood. Blood can flow more slowly along the circulatory system. No, we don't want it to be flowing slowly. We want that blood to be efficiently getting to our tissues. Blood pressure stays the same throughout the circulatory system. Absolutely not. We don't want the same pressure in our lungs and in our aorta, for example. We want high pressure blood in that aorta, and we want lower pressure blood in the lungs, and our veins have an even lower pressure. So definitely not the same pressure throughout. Oxygenated and deoxygenated blood are kept separate. Yes, they are, and that is a definite advantage because then it's more efficient. Oxygenated blood can go off to the tissues and give up its oxygen and collect the waste product, and can come back separately as deoxygenated blood, ready to head to the lungs and be reoxygenated. So 14 is most definitely D. 15. The body has defenses to protect itself from diseases. What is a mechanical barrier to diseases? Okay, so something that stands in the way of those pathogens. So our choices are hairs in the nose, plasma, stomach acid, and white blood cells. Well, plasma not involved. Stomach acid, well, that's a chemical, it's not a mechanical thing. White blood cells, they are cellular. So, mechanical thing standing there in the way, hairs in the nose. Actually stops things entering the body. Okay, so that first line of defense is stopping the pathogens from even getting into the body with that mechanical defense, and it is hairs in the nose in this instance. What comes next? From diseases to exercise. So during exercise, receptors detect a change in the blood and cause the breathing rate to increase. What change do the receptors detect, and where are they found in the body? So during exercise, remember now we have lots of respiration. And at the beginning of exercise, that would be aerobic respiration. And aerobic respiration is giving off CO2. So CO2 is increasing. So the receptors are detecting an increase in carbon dioxide. Where are those receptors sited? Are they sited in the brain or in the intercostal muscles? Now, intercostal muscles are there between the ribs. No, not a chance. Okay, control happening here in the brain. So the change detected is an increase in carbon dioxide, and the site of that detection is the brain. We're going to change the activity of the intercostal muscles, because that's how we're going to change that breathing rate, but it is not where the change is detected. It's detected in the brain. Question 17. An experiment is set up to investigate the uptake of oxygen by germinating seeds. So we've got a whole big setup here, and we need to take the time to make sure that we understand what's going on. So we've got the beakers with colored water. Okay, on the left-hand side, we've got germinating seeds, and they're in a water bath at 25 degrees C. On the right-hand side, we've got dead seeds that are rinsed in antiseptic, so there's nothing living here. Okay, we've got soda lime absorbing carbon dioxide in both sides. So in this case, there's not going to be any output of carbon dioxide affecting any gas level. Okay, so the only thing we're looking at is uptake of oxygen. Now, germinating seeds, again, we are talking about respiration. So we're going to be using oxygen. But it's the living seeds that are going to be using oxygen. So what's going to happen to the levels of colored water at X and Y? Well, at Y, nothing's going to happen because the seeds are there and it's been rinsed in antiseptic. There's not even bacteria in there. Okay, nothing's going to happen. So it's all about what happens on the other side with the living seeds. Now, as those seeds germinate, as they use that oxygen, it's going to pull air out of this little tube to replace this air here. So this line here at X is going to go higher. It's going to rise. So if we look here at the options, X is definitely not going to fall because we're using oxygen. It's going to rise. And at Y, it's going to be completely unchanged. So our answer for this is D. Question 18. The diagram shows a section of a kidney. What are the correct labels? So this is actually just a case of labeling the diagram. So what do we have? X on the outside, making the C shape, is the cortex. Y on the inside is the medulla. And Z coming out from the kidney is a ureter. So X is the cortex, Y the medulla, Z the ureter. The answer that matches is A. Question 19. A boy accidentally touches a very hot object and immediately takes his hand away. Immediately. In this reflex action, what is the effector? So you've got to think back, okay? You've got all of these sort of loops of things that are going on in the nervous system. So you've got receptors, you've got effectors, you've got relays, okay? And where are things happening? Which is your effector? The effector is effective. It gets things done. So when we're talking about the effector, we're saying what made this movement happen? Okay? What made the hand away movement happen? Was it a heat receptor? No, it wasn't. Was it a motor neuron? The motor neuron told the effector to do the job. It did not do the job itself. A muscle in his arm, that would move his hand. The spinal cord? No. Okay, so definitely the effector, the thing that caused the movement, was the muscle in the arm. Our last question for today, question 20. Which description of how the pupil of the eye gets smaller is correct? So we've got the circular muscles and the radial muscles. So remember, if we look at the pupil of the eye, well, the iris, we've got muscles going round like this, okay? Those are your circular muscles. And you've got other muscles like this, radiating out. Those are your radial muscles. Now, in order to make this space here shorter, we need to make these radial muscles longer. So they would need to relax. Okay, and if we want to pull that space closed so that the pupil gets smaller, the circular muscles must contract. Remember, they are opposing muscles. So, circular contract, radial relax. The answer is B. So, in order to make the pupil of the eye smaller, we would need to contract the circular muscles and relax the radial muscles. They're a pair of antagonistic muscles.